Hello everyone, this is Sir ML. Please subscribe to my channel. And in today's video, we are going to delve into the wonderful world of the mitochondria and look at these two processes that work in conjunction with each other to make the bulk of the ATP within mitochondria during cellular respiration. 10 molecules of NADH and 2 molecules of FADH2 that were reduced and formed during the process of glycolysis, oxidative decarboxylation, and the citric acid cycle are oxidized in the mitochondria, and when these two electron carriers are oxidized, they will lose electrons, and those electrons will be transported through a series or chain of protein complexes that are embedded within the inner membrane of the mitochondria. This transport of electrons from one protein complex to the next is coupled with the release of energy that translocate protons from the matrix into the intermembrane space, creating a proton gradient, which will be used to drive the formation of ATP. So now we will take a closer look at how those electrons are being transported. So here we have a typical mitochondrion, and when we zoom in on one portion of the mitochondrion, we will see the two membranes with their own bilipid layers. Here we have the outer membrane, and then here we have the inner membrane, and between those two membranes is the intermembrane space. Towards the inside of the inner membrane we have the matrix, and on the outside of the outer membrane we will have the cytosol. Okay, so now we'll take a look at what happens at the first protein complex, which is also called as NADH-Q oxidoreductase, or complex 1. I'll just name this complex 1 here. Okay, complex 1. The oxidation of NADH to NAD plus will release electrons, which will then be passed on to one of the prosthetic groups within complex 1, which is a flavoprotein, and from there the electrons will be passed on to the 6 ion sulfur centers within complex number 1. The electrons will then move to coenzyme Q or ubiquinone, and the electrons will reduce coenzyme Q into quinol or QH2, which is a mobile electron carrier that is present within the bilipid layer of the inner membrane. Q will be reduced into QH2 after accepting those electrons and also absorbing a hydride ion coming from the oxidation of NADH and a proton that is found within the matrix. At the same time, proton ions will be pumped out into the intermembrane space. Again, complex 1, which is referred to as NADH-Q oxidoreductase, where NADH is oxidized and lose electrons, while Q is reduced and hence gain electrons. Complex number 2, which is also known as succinate Q reductase, has its own prosthetic group FAD and also its own ion sulfur centers. In the citric acid cycle, the oxidation of succinate to fumarate by the enzyme succinate dehydrogenase produces FADH2, and the oxidation of FADH2 into FAD will produce electrons that will be passed on directly into the iron sulfur centers of complex number 2. From there, the electrons will move into again the mobile protein quinone, and again Q will be the final acceptor of those electrons at complex 2, and hence Q will be again reduced into QH2. Complex 2 is the only protein complex in the inner mitochondrial membrane that cannot pump protons out into the intermembrane space, and that is why the oxidation of FADH2 produces much less ATP as compared to the oxidation of NADH. QH2 will be then repeatedly oxidized and reduced at complex 3, which is also known as Q-cytochrome C oxidoreductase, and will pass its electrons into the prosthetic groups of complex number 3, which is cytochrome B and cytochrome C1, and the final acceptor of those electrons will be cytochrome C, which is another mobile protein electron carrier that is present in the intermembrane space of mitochondria. So in this step, QH2 will be oxidized and those electrons will be passed on into the cytochrome B and cytochrome C1 of complex 3, and the final acceptor of those electrons at complex 3 will be cytochrome C. So therefore, cytochrome C will be reduced. In the next step, cytochrome C will be oxidized, so cytochrome C will now lose those electrons and pass those electrons into the cytochrome A and cytochrome A3 of complex number 4, which is also referred to as cytochrome C oxidase. Because why? Because cytochrome C is now oxidized, and remember, oxidation is losing electrons while reduction is gaining electrons. The prosthetic groups of both the cytochromes in complex 4 will be reduced, and the final acceptor of those electrons will be molecular oxygen. It will accept those final electrons. So oxygen will receive those will accept those electrons plus protons from the matrix and be reduced into water. At the same time, when these electrons are being transferred from these two complexes, more proton ions will be passed on into the intermembrane space. So there will be a build-up of protons in the intermembrane space, which makes the intermembrane space positively charged while the matrix become negatively charged, and hence there is a concentration gradient. And in nature, when a gradient is produced, everything wants to balance itself out, everything wants to maintain homeostasis. So these protons will want to move back into the matrix to try and maintain equilibrium between the intermembrane space and the matrix. So they will do this through facilitated diffusion, and that is, they will move through this very special protein complex and sort of move and mechanically rotate this protein complex, which is referred to as the F1 F0 ATP synthase. The F0 part will be embedded in the inner mitochondrial membrane, while the F1 knob will protrude into the matrix. So this movement of proton ions through this protein will physically jam ADP and a free phosphate group to make ATP, and this happens at the beta subunit of the F1 knob. So to summarize, the oxidation of NADH and FADH2 releases electrons that will be passed on through a series or chain of these protein complexes, and this transportation of electron releases a lot of energy. That energy is used to pump out protons from the matrix into the intermembrane space through these protein complexes or protein pumps to create a concentration gradient. The concentration gradient will be used to power the motive force of the proton ions from the intermembrane space back into the matrix, but this time they will move through a very special multi-subunit protein complex known as F1 F0 ATP synthase will cause the phosphorylation of ADP with a free phosphate group to make ATP. The oxidation of NADH molecule produces around 2.5 ATP, while the oxidation of FADH2 produces 1.5 ATP per molecule of FADH2. So in total there will be 10 NADH and 2 FADH2. So the total will be 25 ATP from NADH and 3 ATP from FADH2, which brings the total to 28 ATP molecules. And when you add those 28 ATP molecules plus the 2 ATP molecules we get from glycolysis plus the 2 molecules of ATP we get from the citric acid cycle, we get the magic number of 32 ATP molecule per molecule of glucose. Again, the oxidation of NADH and FADH2 produces electrons that will be transported through a chain of protein complexes, and this transport is coupled with the translocation of protons from the matrix into the intermembrane space that creates a proton gradient, and that gradient will be used to phosphorylate ADP and P to make ATP. So that is all about the electron transport chain and oxidative phosphorylation. I hope you found this video helpful, and I hope now you know why mitochondria is referred to as the powerhouse of the cell. Thank you.